Hello, this is Alice Formiga of eOrganic, and I'd like to welcome you all to today's webinar. I hope very much that you and your families are healthy, and we're very grateful that we can continue to provide our webinar program. So today, we're going to learn about the microbiome, what it is, and how it might impact organic dairy production. eOrganic publishes free articles, videos, and webinars about organic farming and research, and you can find all of them on our new website at eorganic.org and on the eOrganic YouTube channel. So today, I'm very pleased to welcome our presenters, who are a team of researchers working on a project funded by NIFA OREI. The project leader is Dr. Noelle Noyes of the College of Veterinary Medicine at the University of Minnesota. And she will be joined by others on her team, Dr. Bradley Heinz, Dr. Luciano Caseta, and Christopher Dean, also of the University of Minnesota, as well as Dr. Vicinius Machado of Texas Tech University, and possibly also Pablo Pinedo of Colorado State University. So without any further ado, I'm going to hand over the screen controls to Noelle Noyes. So,
can you hear
okay,
me, Alice?
you've got it. Yeah, you've got it now.
Okay, and you can hear me?
Yep, I can hear you.
Great. Okay, I am just going to back up the slides here for just a second. So thanks everyone for attending the webinar, especially these days with everything going on. This is our first webinar that we're giving as part of the USDA funded grant that Alice mentioned that focuses on the microbiome and mastitis. Before I get started into the nitty-gritty of the presentation, just a couple of sort of logistical notes. The first being that if you hear background noise coming through my audio, I do apologize for that. We do have young children and various animals in the background, and my husband is pretty good at wrangling them, but not always 100% perfect. So apologies for any noise that comes through. The second thing I want to do is just take a moment to acknowledge that many of you on this call, I would assume, are probably continuing to do your part to produce food during the coronavirus epidemic. And I think that we can only imagine that that's probably extremely challenging given labor challenges and other constraints. So just a big thank you from our project team to everyone on this call who is continuing to ensure that we have safe and secure food during this time. With that, I will turn to a less urgent topic, but one that we focus on a lot within our research team, and that is the microbiome. I will preface the presentation by letting you know that this is definitely going to be a high-level presentation about the microbiome and its potential impact on dairy production. It's a little bit different than maybe some webinars in that we won't be providing detailed research results yet in this talk, but rather what we're trying to do is set the stage for what we believe is going to be a big wave of microbiome research results that will be coming out over the next several years, not just from our group, but also from many other research groups who are involved in agricultural research and who are now integrating microbiome into their research programs. And so what we have tried to do as part of our USDA funded work is to set a goal that to make sure that this microbiome science is channeled into applied on-farm benefit. And so what that means is that basically trying to bring the science into something that's directly impactful to all of you as people who are involved in producing and supplying food. Part of that means creating awareness around the microbiome and trying to engage in conversation with all of you about how microbiome research can best support improvements in dairy health and production, and specifically organic dairy health and production. So with that, I will echo what Alice said, which is that the work that we'll be talking about today and that we're doing is made possible only by a team of dedicated researchers who are listed on this slide. So while I'm going to be giving sort of the formal part of this presentation, all of these researchers are also available on the webinar to answer questions about any of the work. And it does take a diverse team of researchers to conduct microbiome research that is applied. And so I want to quickly list sort of the expertise of each person on the team. So Dr. Brad Hines is an organic dairy specialist, and he is in the animal sciences department at the University of Minnesota. And he manages and runs an organic dairy herd out at the Morris Research and Outreach Center, which is in Morris, Minnesota. So he has hands-on experience with organic dairy production, and then also has been incorporating microbiome science into his work. Dr. Luciano Caixeta is also at University of Minnesota, and he is a veterinarian and also a PhD researcher who specializes in metabolic diseases of dairy cows and dairy production. Myself, Noelle Noyes, I am also a veterinarian and an epidemiologist, and I focus on bringing microbiome research to issues of livestock production, including organic dairy production. Dr. Pablo Pinedo is an assistant professor at Colorado State University who specializes in dairy management systems. And Vinicius Machado is an assistant professor of dairy health and management at the Department of Veterinary Sciences at Texas Tech University. And then you all probably know Alice Formiga, who is assistant professor of practice for eOrganics at Oregon State University, and is really helping us try to bring the microbiome research to all of you. So with that, the work that before I get to that, the next thing I want to say is that we have many students and a couple of postdoctoral researchers involved in this research, and they are doing a lot of the on-the-ground field sampling, lab work, and then the bioinformatics and statistics needed to do microbiome research. And so I think we've had nearly 20 various vet students, undergraduate and graduate students involved in this project already, helping us with the work. And obviously we couldn't do this work without their help. A couple of them are on the webinar today and will also be helping to answer questions. So the work that we're talking about in this webinar itself is supported by a grant from USDA, and specifically the Organic Agriculture Research and Extension Initiative, or OREI. So the project has a very long title, but I will read it because it's actually very descriptive in terms of what we're trying to do. So the title is Catalyzing an Open Community Research and Education Program to Leverage the Microbiome for the Advancement of Organic Livestock Production Using Mastitis as a Test Case. So basically what we hope to do is we think that there's a lot of power in the microbiome, and we want to open up that power to multiple researchers and education and extension specialists across the U.S. agricultural community, so that we basically sort of seed research and development within the microbiome. And so all of the work that we're doing is open source. That includes the data and all of the analytical pipelines that we're putting together. And then we hope to make that available to producers on the ground. We believe that the microbiome is going to be useful for all organic livestock producers, but we are specifically interested in dairy production in this project. And what we hope to do is use mastitis as sort of a test case or a use case scenario to show how the microbiome can be leveraged, hopefully for prevention and therapies for mastitis. We do have a website hosted by eOrganic. We hope to put more updates on the website as the project, as results start rolling in on this project. And so please definitely take a look at the website and check back for research updates and outreach and extension updates as they happen. So the plan for today, I've broken the presentation up into 4 different areas. So the first is to give, again, this high-level overview of the microbiome. I'm sure many of you on this webinar are familiar with the term microbiome, but it can mean different things to different people. Some people think of prebiotics and probiotics as a type of microbiome therapy, and indeed they are. And so what I want to do is spend a little bit of time talking about what the microbiome is, so that everyone's sort of on the same page in terms of what we mean by that term. The second section will be overviewing key microbiome concepts and how those translate to dairy production. The third will be talking about one of the many challenges of doing applied microbiome research, and I think this will help everyone on the call appreciate sort of the challenge that we're up against, but also how we can tackle it. And then the last part, we'll talk about what might the future bring in terms of microbiome research for applied organic dairy production. So to get started, first, the high-level overview of the microbiome. So before I get into the what is it, I want to talk about why the microbiome. So the USDA and other funding agencies and companies and individual farmers are actually dedicating a lot of financial resources right now to microbiome research. And the reason why people are so interested in the microbiome is because the microbiome is really crucial for not only dairy cow health and production, but basically every living thing on the planet. And that includes things like soil health, crop health, all livestock health and production, and then of course human health and production. So the microbiome is just fundamental to health and production across all sorts of areas. And so now that we have the research tools and the scientific technologies to access the microbiome, the next step is of course to start generating a lot of knowledge about the microbiome. And that's sort of where we are in agricultural research right now, is trying to build our knowledge base about the microbiome and how it specifically relates to various challenges and concerns in dairy cow health and production. So what is the microbiome? I boil it down to, it's the invisible universe in, on, and around us. And I think that really captures it, because just like the universe and all of its complexities that we can see with our eyes, there's also an even more complex and more extensive universe that we can't see. And it's right now coating our bodies, it's coating the insides of our bodies, and it's coating all of the surfaces that we touch around us on a daily basis. And so rather than try to put together a bunch of stick figures about the microbiome, I want to try to use a YouTube video that was put together by NPR that talks about 4 minutes and gives a really nice overview of the microbiome and where we are in the science of it. It took a while to find a video that I thought was both scientifically grounded and also digestible. So hopefully that was helpful. So I think it gives a good overview of the microbiome. And now I want to refer back to that video to talk about some key microbiome concepts, and again, how they translate to dairy production. So, can everyone hear me there? Alice, can you hear me?
Yeah, I hear you. Yeah, that skipped out for a second, but it's okay.
Okay, I had a little warning that my internet connection is unstable, so hopefully we're good. I know. Okay, so the first point that I want to talk about is that, as you saw in the video, the microbiome sort of starts at birth and then changes with age or development. And so in humans, we think that the microbiome is extremely dynamic in the first few years of life, and then sort of starts to stabilize, and then actually is very resilient to change after that initial development phase. And so that's sort of the trend that we see within humans. And actually there's been some nice research in dairy calves that the same thing happens in calves as well. So this is the first plot that I'm going to show you that's called an ordination plot. And these ordination plots are very common in microbiome research. And so I do want to spend some time walking you through sort of how to read one of these figures, because you may see them over and over again as more and more microbiome research comes out. So on this ordination plot, each symbol is a sample that was collected from a dairy calf. And so the symbol is basically representing the microbiome in that sample. So all of the bacteria, potentially viruses, and other types of microorganisms that were in that sample. Symbols that are close together on this figure have similar microbiomes, and symbols that are far apart have very dissimilar microbiomes. So you can see the symbols that cluster together are ones that came from calves that have similar microbiomes. In this figure, each symbol is colored by the week of age at which the sample was collected from the calf. So the red symbols were all collected from calves at 1 week of age. The orange from calves at 4 weeks of age, yellow at 8 weeks of age, green at 1 year of age, and blue at 2 years of age. In this study, they also fed these calves different diets, and that's why you see different shapes of symbols, so triangle, circle, square. So what you can see here is that the red dots or symbols are sort of clustered together on the upper left-hand corner of the figure. And then we move to the orange dots, which are clustered sort of in the middle there over on the left-hand side. And then the yellow dots are clustered together at the bottom. So what you see here is, just as in humans, dairy calves, the microbiome of dairy calves basically develops over time. And so you see this sort of transition in the microbiome composition between 1 and 8 weeks of age, and it's very dynamic. We don't know what bacteria are sort of increasing and decreasing within the microbiome. So this plot only shows us sort of the differences between the different samples. But what we can say is that the microbiome is shifting very dramatically during those first 8 weeks. Then when you go back and look at the microbiomes of these calves at years 1 and 2 of age, you can see here over at the green and blue that the microbiomes at 1 year of age, they're very different from what the calves had at 1, 4, and 8 weeks of age, but they're very tightly clustered together. So that green blob there is all the samples tightly clustered together. And then the blue samples at 2 years of age are, again, very close to the green samples and very tightly clustered together. And so what that means is that by 1 or 2 years of age, the microbiomes of these calves have really sort of stabilized. And so you're not seeing like a lot of dramatic change in the microbiomes at that 1 year or 2 year of age time point. So just as in humans, we see that calves do experience this rapid evolution of their microbiomes in the beginning of life, and then that stabilizes maybe by 1 or 2 years of age. So that's really important to know, because if we're looking at adult cows, like lactating dairy cows, then potentially we're dealing with microbiomes that are very stable and potentially harder to sort of manipulate if we want to. Whereas with dairy calves, you know, if we want to understand how we can improve the microbiome of a dairy calf that is within the first 2 months of life, we really need to be specific about what age we're targeting, because the microbiome of a 4-week-old calf looks different than the microbiome of an 8-week-old calf. So we have to be careful in our study design and in how we're interpreting results of our research based on the age of the animal that we're working with. The second important point from the video that I want to talk about is this concept that each person has a unique microbiome fingerprint. So in the video, it talked about different individual humans having unique microbiome fingerprints by the time they've reached sort of adulthood. We do see that in animals as well, in livestock animals, although it's interesting. So I do work across species, and in swine production especially, we see very strong fingerprints of microbiomes within litters of piglets. So there seems to be this sort of unique microbiome fingerprint for each piglet sow litter that we see in swine production. In dairy cows, of course, one dairy cow typically gives birth to one calf, and so we don't really have the opportunity to see that sort of clustering of fingerprints within dairy cows. But what we do see, and what we've started to see, is that actually dairy farms can have their own unique microbiome fingerprint. So on this next slide, again, this is another ordination plot. It looks a little bit different. So on this plot, again, each dot is a sample that we've collected from one of 3 organic dairy farms across the US. And each sample represents, each dot represents a sample that we collected on that farm. And again, dots that are closer together have more similar microbiomes. And we've colored the dots by the farm at which the sample was collected. So red is farm 1, green is farm 2, and blue is farm 3. So what you can see here again is that the green dots, farm 2, sort of cluster together at the top of the graph. The blue are sort of clustered together at the bottom left, and the red are clustered together on the bottom right. And so again, this goes to the concept that different farms sort of have their own unique microbiomes. And this has implications for research, study design, and also research results. So if we're designing a study about the microbiome, we need to keep in mind that, you know, we're going to have these farm level effects. And so therefore, we may want to sample multiple farms so that we're sort of getting a representative view of the microbiome across farms. On the flip side, if you see that a microbiome product has been tested on one farm, let's say as an example, and it produced really good results, it may not be the case that that microbiome product will work equally well across all farms. Again, because farms tend to have, it seems like, their own unique microbiome fingerprints. Now, some of these, there are overlaps in the microbiomes between these farms, and there are things that are consistent and common across farms. So we can't automatically say that, you know, a finding in one farm is not applicable to another farm, but it's just something to keep in mind that we can't automatically say that research results in one farm are reproducible across other farms. Does each data point represent, I'm just reading the questions. Each data point represents one sample taken from that farm. And in this is preliminary data. So each dot could be, multiple dots could be from one animal. They could be from one sampling day on that farm. And actually I have another slide after this to talk about the effective sampling day or year. So that's a great question. Again, this is preliminary data, and these are samples that we're processing actually right now. Not right now because our lab is shut down due to the coronavirus, but we're processing these samples on an ongoing basis. So these are the first batch of data points that we received back from the sequencer when we process these samples. So we have this idea that farms each have their own unique microbiome fingerprint. Also, I will say that these farms are not geographically close to each other. They are across different states, actually. So that's another great question that I received in the comments. And so again, region or geography could be playing a role in this, but it's also something to note that if, you know, something comes out about the microbiome from farms in Pennsylvania, that may not be applicable to farms in, say, Minnesota. So not only do microbiomes sort of fingerprint by farm, but they can also fingerprint by sampling day or time. So this is these are some nice figures from John Barlow's group out of Vermont. And this basically shows the impacts of sampling month and year on the microbiomes that these researchers sequenced. So on the left-hand side on that chart, these researchers went into a single dairy farm in Vermont, and they collected both milk samples and also teat apex skin samples from cows that had mastitis and did not have mastitis. So that would be healthy versus infected. And they did this in years 2015 and 2016 in August, July, and June. So a monthly sampling time point. And the first thing they did when they looked at the data is they looked at how the microbiome may have varied by sampling month. And so what you can see is that indeed the microbiome composition tends to sort of cluster by sampling month and year on the left-hand side. On the right-hand side, they've done the same thing. So they've taken the microbiome data, and each sample is represented by a dot on the picture. And they've demonstrated that the microbiome found in the milk samples is different than the microbiome found in the teat epithelial or skin samples. So that makes sense. The milk microbiome is different than the teat skin microbiome. But in addition to that, they also looked at how years 2015 and 2016 samples may have differed from each other. And so the 2016 are in the black dots and the 2015 samples are in the open circles. And what you can see is that the 2016 milk samples cluster much separately from the 2015 milk samples, and same for the teat skin samples. So again, this is just showing us that there is microbiome sort of clustering or fingerprints both by farm and potentially by time of sampling within farm. And so what this means is that we have to account for this variability when we're looking at microbiome in research studies and applied microbiome science. The third important point that I want to talk about is that within an animal, there are different microbiome ecosystems. So in the video that we watched, they talked about sort of the wet microbiome, so like the mouth, the armpit, etc. And they talked about oily microbiome, like the back, the skin on our backs, and then dry microbiome, like the skin on our forearm. So those are different microbiome ecosystems in the human. There are also many microbiomes within the lactating dairy cow. And so you can imagine that there are different physiological and anatomical compartments within the dairy cow. So people have looked at the respiratory microbiome. They've looked at the, for instance, vaginal and uterine microbiomes. And of course, they've looked at the gut microbiomes. And so this slide right here is a nice article published by Yeoman et al. In 2018. And in this, what they did is they really went into detail in the different areas of the gastrointestinal tract. So they started at the pharynx, and then they went through the reticulum, rumen, omasum, abomasum, and then they went into the small intestine and they looked at duodenum. They looked at 3 different sections of the jejunum, then they went into the ileum, the colon, and then feces. And so I want to draw your attention on the ordination plot to the green dots on the far left side. So those are the microbiome from samples taken from the proximal jejunum. And you can see that even though the proximal jejunum is positioned between the duodenum, which is the light green, and the medial jejunum, which is sort of the teal colors, that the proximal jejunum actually has a much different microbiome than those other close by sections of the gastrointestinal tract. I have a question about the scatter plots. Is there corresponding X and Y axes? Does the position on the plot correspond to the kind of bacteria it represents? Great question. So the X and Y axes on these plots are sort of, I'm not going to say that they don't have a lot of meaning, but they're basically a measure of distance of how far apart these samples are from one another based on their microbiome composition. So it's not a type of bacteria, it's all of the different bacteria put together and their relative abundances within each sample or within each dot. So again, it's an overview of the composition of the microbiome, not looking at individual bacteria and how abundant or not they are. And so it's basically trying to take the thousands of different bacteria that you find in each of these samples and each of these dots, and using that information to say how different are these samples from one another based on the distribution of those thousands of bacteria across each sample. So hopefully that helps. So as you can see here, those green dots on the left-hand side from the proximal jejunum, they have microbiomes that are very similar to one another. All those samples were collected from the proximal jejunum, and the distribution of all the thousands of bacteria in those samples are very similar between those proximal jejunal samples. If you look down at the bottom of the chart where you have the open green circles, those are also from the proximal jejunum, but they're clustering very far apart from the proximal jejunum samples on the left-hand side of the chart. And that's because those open circles were taken from the mucosa of the jejunum, meaning the researchers scraped the walls of the jejunum and took the bacterial communities that were like really tightly adhered to the jejunum wall. Whereas the samples on the left-hand side that are filled in green, those are basically the contents of the jejunum. So the contents that are in the lumen or the middle of the jejunum. And so what I wanted to point out here is that, even just looking at the bacterial community that's adhered to the walls of the jejunum, that is very different. The ecosystem in that tightly adhered content is very different than the microbial community that you see within the contents of the jejunum itself. And so when we talk about sort of a dairy cow microbiome, you have to be a little bit more specific about what you're talking about. Are you talking about the gastrointestinal tract? And if you are, what area of the gastrointestinal tract? So a lot of studies, including ones that we've done, we look at the fecal content because it's easy to access, right? You can easily get a fecal sample from a dairy cow. It's much harder to get a jejunum or a duodenum sample from a dairy cow. You basically have to sacrifice the animal to get that. So there's a lot less research about the duodenum, jejunum, etc. of the dairy cow, when in reality, those microbiomes may be more important for things like calf scours or other types of metabolic diseases of dairy cows. Those communities may be more important to research than the fecal bacterial community. So This is a challenge when it comes to microbiome research, but the good news is that we understand that there are different microbiomes across the dairy cow, and we know that we need to be specific when we're talking about microbiomes of dairy cows. Oh, let's see. I can't, there we go. Okay, so what about the mammary microbiome? So in this USDA funded project, we were specifically interested in trying to understand whether or how the mammary or udder microbiome plays a role in mastitis. And so there's definitely more literature out there on the gut microbiome and the respiratory microbiome of cattle, but there is some really intriguing literature about the microbiome and potential role in mastitis. So I'm going to walk you through one of the first studies that came out looking at this potential relationship. So this is a study that was done in just some dairy cows who were sampled when they were all healthy in terms of mastitis. So the sampling was conducted when there were no signs of clinical or subclinical mastitis. However, some of the cows had a history of clinical mastitis, and those are denoted in this chart with the red dots. So basically MQ means a quarter with a history of mastitis. So QM, quarter with mastitis. The black dots are quarters that had no history of mastitis. So they were confirmed to have always been clinically normal in terms of mastitis. The green dots are for quarters that were sampled that they weren't able to determine whether those quarters had a history of mastitis or not. And so what you can see, this is again another ordination plot with sort of meaningless axes. The red dots cluster separately from the black dots. And so what this is telling you is that quarters that had a history of mastitis tended to have a different microbiome than quarters that had always been healthy. So this was the first sort of evidence that we had, or one of the first pieces of evidence that the scientific community had, that somehow the microbiome may be playing a role in mastitis. More recently, John Barlow's group at Vermont came out with this really interesting study. So this plot is a really interesting plot. It's a bit busy, and it's a bit different than the ones that I've been showing you before. So let me walk you through it. So in this figure, each dot is not a sample anymore. Now it's an actual type of bacteria. So each dot represents a type of bacteria. And again, the dots are clustered by whether or not they were found in healthy or infected milk, or healthy and infected teat skin. So again, these samples were taken from cows that either had subclinical or clinical mastitis or were healthy. And so what you can see if you look over on the left-hand side at the milk results. So the healthy milk samples contained a fair number of bacteria represented by that far left cluster. The healthy milk contained many types of bacteria that were not found in the infected milk. So the infected milk is further to the right there. And then there were some bacteria in the middle that were found in both healthy and infected milk. But actually, not very many bacteria were common to both healthy and infected milk. And so what you're seeing here is that there are bacteria that sort of uniquely identify infected milk, and there are bacteria that uniquely identify healthy milk. So there's a big separation between the microbiomes, that the microbiome profiles you see in milk from healthy versus infected glands. On the right-hand side, they're showing the results for samples taken from the teat skin. So the teat apex was swabbed basically, and then that sample was subjected to microbiome analysis. So what you can see is that there's a huge sort of glob of circles in the middle. And so what that's telling us is that many microbes, so there were thousands of microbes found in these samples, and many of those microbes were found in the teat skin from both healthy and infected glands. So that's interesting because it tells us that regardless of whether or not the cow had mastitis or not, many of these bacteria were there regardless. But you do see that there are some bacteria that were unique to the teat skin from healthy cows and unique to the teat skin from infected cows down on the bottom. So you do have some differences between teat skin microbiomes of healthy and infected cows, which is also potentially important information. Potentially the bacteria that you see in the healthy teat skin, those may be protective against mastitis, as an example. So this is where we can start generating some hypotheses about how the microbiome of these different compartments, both milk and the teat skin, how they may play a role in mastitis. So this was a paper that just came out in 2019, and as you saw in the video, much of this research is very new. So 2016 is actually considered sort of older research when it comes to microbiome. So things are just accelerating at a really fast pace in terms of research results. And this is a very nice study demonstrating that there likely is a role of the microbiome in mastitis. So those are some of the key microbiome concepts and how they translate to dairy production. I want to sort of start with those results that I just showed you and talk about one of the many challenges of doing applied microbiome research. So one major challenge that we have when it comes to the microbiome is trying to sort of decide what's the chicken and what's the egg. And so the results that I just presented to you, those results are what we term cross-sectional largely. So what they did is they went into a population of dairy cows, and they sampled those dairy cows both for mastitis and then also for their microbiomes at the same time. And so then they go back and they say, okay, the microbiomes of cows that had mastitis look different than the microbiomes of cows that did not have mastitis. And so that's very interesting information, but what we can't really say with a lot of confidence is what came first. So did the microbiome sort of become disrupted in some way, and that then led or contributed to the development of mastitis? Or did the cows get mastitis for some reason unrelated to the microbiome, and then because there was this invasion of pathogen, then the microbiome became disturbed, and therefore there was a different microbiome in the mastitic cows? So this chicken and the egg problem is really important for us to tease apart. Why is it important? Why does it matter? Okay, I'm going to walk you through why it matters, whether or not microbiome happens first or pathogen happens first. So in scenario number one, we have a microbiome, what we call dysbiosis. So basically a huge disruption to the microbiome leading to pathogen invasion. So on this chart, the x-axis is time. So this is just time in a dairy cow. And on the left-hand axis, we have what I term microbiome health. So the green line, if you're high up on that left-hand axis, that means that this cow has a quote-unquote healthy microbiome. If you go down on the left-hand axis, you start getting into an unhealthy microbiome, and eventually you reach a term, or the cow reaches a term called dysbiosis, where the microbiome is just considered abnormal. On the right-hand side, we have what I call pathogen activity axis. So the higher up you are on the red, the more pathogen activity there is in terms of a mastitis pathogen within the mammary gland. So in this scenario number one, let's take a dairy cow. The dairy cow starts out on the left-hand side with really nice microbiome health of the mammary gland and the udder. So high microbiome health and low pathogen activity, right? So clinically healthy and subclinically healthy cow. Over time, for some reason, this cow's microbiome starts deteriorating. Maybe there was an unrelated health challenge. Maybe it was parturition, right? So this cow gave birth and that led to some sort of disturbance in the mammary microbiome. So the microbiome health starts deteriorating and eventually sort of deteriorates into full-blown dysbiosis. At that point, that allows for increased pathogen activity because the defense, that first line defense of the microbiome is disrupted to a large degree. So then you see this increase in pathogen activity, which then can lead to mastitis. So this is scenario one, where the microbiome comes first and mastitis comes second. Let's contrast that to scenario number two. So in this scenario, again, you're starting off on the left-hand side with a cow that has high microbiome health and low pathogen activity. So this is a good scenario. But in this scenario, the animal experiences an increase in pathogen activity. Maybe there's an environmental exposure. Maybe there's there's a loss of integrity of the mucosa of the teat canal or something that allows this pathogen to invade and start replicating in the mammary gland. So you see the red sort of, the red curve spike up. that causes, in turn, a deterioration in the microbiome. So that's when you start to see this deterioration of the microbiome going down, down, down until you get into this dysbiotic state where the microbiome is abnormal. So this is where pathogen invasion leads to a microbiome dysbiosis. Now, in reality, scenario number 1 and 2, we could have a little bit of both going on, right? And that's because biology is complicated, that's probably the case. But you can see how these two different contrasting scenarios can make a big difference. And I will say that they make a big difference because the answers to whether or not we're dealing with scenario 1 or 2 are going to dictate the efficacy, type, and timing of any microbiome-based interventions that we want to use. How does that happen? Well, let's take scenario 1, right? So you remember the scenario where maybe the cow gives birth and the microbiome is disturbed and there's this dysbiosis. Maybe we intervene with a microbiome-based therapeutic right when we realize there's a disruption to the microbiome, or we suspect that there's a disruption to the microbiome. That's when we give our microbiome-based intervention with the little lightning bolt. Well, that might allow us to do this, right? So we might be able to completely change the outcome of mastitis in this animal because we support microbiome health at that critical juncture. That helps the microbiome rebound. We see a little uptick in pathogen activity, but we've intervened with the microbiome enough to then suppress pathogen activity. So that would be a really great scenario in terms of using microbiome to prevent mastitis from ever occurring. If we take scenario 2, where this cow is experiencing increased pathogen activity unrelated to the microbiome, you may see the clinical manifestation or subclinical manifestation of the pathogen in terms of mastitis. And then you say, okay, I'm going to intervene with some sort of targeted microbiome, maybe even like a microbiome transplant. I'm going to take a healthy microbiome and I'm going to give it to this cow because I know that they're experiencing a full-on onslaught to their microbiome. So I'm going to sort of repopulate them. And this is what we see in humans with C. difficile as an aside. So we repopulate that microbiome. And by doing that, we change the dynamics of the pathogen activity. So we support the microbiome that will help sort of weed out the pathogen, and then the pathogen then goes down much more quickly than it otherwise would. So this maybe helps us change the disease pattern. So these types of details are really important, but they're really challenging to discover because it takes intensive sampling and other things. This is just one challenge that I've talked about. There are also many other unanswered questions that we need to get a handle on in order to basically come up with really applied, effective, robust microbiome preventives and therapies, which is the ultimate goal, at least of our research. So what might the future bring, given all of these challenges and all of these opportunities? The good news is that tons of research is being funded and conducted. So the project that we're doing right now, we're sampling over 600 heifers in organic dairy operations, and we're following them over time. And studies like ours, there are many studies being funded and conducted right now related to microbiome research and dairy health and production. And so I think that that's really good news because it means that within the next few years, there's going to be hopefully sort of just an explosion of these research results, and we're going to actually start seeing some applied benefit. We've also seen lots of interest from farmers, producers, veterinarians, and others in this. And that's really important because it's important for us to conduct these studies in commercial operations, in actual farms, where we can make sure that we're representing the microbiomes that are actually happening on farms. And so this has been great, and it's a great area of support for us. There's also lots of untapped potential. So you saw in the video that I showed that microbes do all sorts of things. They regulate the immune system. Some microbes pump out sort of natural, quote unquote, natural antibiotics that kill other microbes. And so that latter point is something that we're very interested in as a potential therapy for pathogens. So can we find these compounds produced by a bacteria that can selectively prevent pathogen invasion, as an example. So there's tons of untapped potential within the microbiome, which is also the good news. So us specifically on our project team, how we're trying to help is that we are trying to tackle this chicken and egg problem and really represent the temporal dynamics of how the microbiome and mastitis interrelate over time within lactating dairy cows. So we are undertaking time series intensive sampling with the help of farmers across the US to try to sort of replicate those curves that I showed you. And that we're probably generating about 12,000 samples in order to do that, and that's with the funding from the USDA. So stay tuned on those results. We also hope to use that data to identify bacterial-derived compounds that may prevent or treat mastitis. So that's what I alluded to on the previous slide. So again, stay tuned for that. We are also hoping to generate more educational materials that sort of explain microbiome research and ensure that we are tying our research and our science to applied uses and microbiome-based discoveries. And so this webinar is an example of that. Our openROAMER website will continue to have updates and sort of educational materials posted to it, and we will have more webinars that will get into things like pre and probiotics. We'll start sharing research results with you as we generate them. And then finally, open source data and research pipelines. So this is a really important area for us and one that we're really dedicated to. Microbiome research can be a steep learning curve for researchers, scientists, extension agents, etc. And so what we're doing is making all of our microbiome data open source, and we're putting it into a really usable research pipeline so that other researchers and other scientific groups can use their subject matter expertise and integrate the microbiome into that, and do it in a way that's more usable and really involves less expertise in coding and bioinformatics and computer science. So stay tuned for that. Our graduate students are working really hard to make that resource really nice and usable and accessible. So with that, I again want to just give you the information about this grant. We're really happy to have the USDA supporting these types of foundational studies in the microbiome. I really thank you for your time, especially during these crazy times, and we really would love to answer any and all questions that you have. I tried to answer them as we went, but I think I may have missed a couple. Again, here are the people on the call who are available to answer questions.
Okay, thank you, Noelle, for that very clear and really fascinating introduction. We're going to start our question and answer period. So if you have a question, you can just type it into the Q&A box on your screen. You can also type it into the chat box, and we'll be reading the questions out loud until we run out of time or run out of questions. If you don't see those boxes, you can hover over the bottom of your Zoom screen and some controls should pop up on a black bar. And if you click on the Q&A one or the chat one, that should bring it up. So I just wanted to mention again that this presentation has been recorded, and you'll be able to find the recording in approximately a week, maybe 2 weeks at most, given all the craziness going on the eOrganic YouTube channel. We'll also be sending you a follow-up email survey later today, so we'd really appreciate your feedback with that. So moving on to the questions here, let me open up those boxes and make sure. Oh yeah, this one came in during the webinar, and I don't think we talked about this one. Are you taking soil samples as well to see how soil biology composition might correlate with the cow's microbiome? And this person is also very interested in the relationship to feeding and grazing practices to the microbiome.
So I've talked a lot, so I want to open it up to any of my project collaborators if anyone's interested in taking that question.
Okay, you can unmute your mic, sir.
Okay, I can answer it. So we are not taking soil samples as part of the project that we're doing. I will say I'm very confident that feeding, diet and grazing practices will impact the microbiomes of dairy cows. I think the gut microbiome and potentially also the skin microbiome. So definitely that's an area that is very is very pertinent and relevant. I do know that there are other research groups working on things like bedding, pasture-based systems, and how that impacts mastitis. And then there's also a microbiome component to that. So looking at sort of the interrelation between bedding management, mastitis, and microbiome. So yep, a great question. I'm sure that that's quote unquote a confounder or something that we could include in the study, but we have to make decisions in these studies about what confounders we're going to include and which ones we're not. And so in this study, we're reporting sort of the conditions and the management strategies in these different farms, but we're not actually taking soil samples.
Okay, and if anyone else that wants to chime in, go ahead.
So this is Luciano Caixeta from University of Minnesota too. And then just to tie in, there's a question on the Q&A box about like, would you be able to sample soils like we do for microbiome, like we do for other things now? And definitely there's an opportunity to do that, like as long as things are done in a proper way. As a matter of fact, like we have been working on putting videos and directions on how to collect sample milk and or teeth skin samples for microbiome. So that would be a matter of like collecting properly, following like good quality collection methods, and that could be done. Said that, I'm definitely not a soil specialist, and that will be something for a soil specialist to answer and see how you can collect that and how would you use it.
Yep.
Okay.
There's definitely a lot being done in soil microbiome as it relates to crops and also feed. So it's a huge area of research right now.
Okay, here's a question about how abundant bacteria are deep in the mammary gland of the cow with no mastitis. I know you showed the figure that showed the microbiome of milk and the microbiome of teats, but this person's asking about how it is deep in the mammary gland.
Again, I will open it up to anyone on the panel who wants to answer.
Okay. So this is Vinicius Machado from Texas Tech. I'm not particularly very knowledgeable on this matter on like the deep mammary gland, like in the alveoli. For example, in this question here, so like, is the mammary gland sterile at the level of milk secreting cells, alveoli, like we have been taught? And I'm gonna put this on perspective on the uterus as well, because like, during vet school, I was taught that the uterus was a healthy uterus was a sterile environment. And now with all this microbiome work that is being done, we now know that even pregnant uteruses are not sterile. So even within the placenta of dairy cows or beef cows, there are some bacteria that it's living there. Not necessarily living, but they you can detect DNA from those bacteria in those samples. So my guess is that if we do a deep sampling in the mammary gland, like in the alveoli level, I guess we're going to find bacteria there. Most likely is that the profile of the microbiome is going to be very different from a mammary gland that is inflamed with clinical or subclinical mastitis, but I believe there will be bacteria there.
Okay.
Luciano, do you want to talk about the sampling that you just did?
Yes, so we are actually trying to look into that by collecting sample like straight from the gland cistern using transcutaneous with needles without going through the teeth canal and the teeth end to see if we can find some of the microbiome there. One thing that like, and we just collected it like right before they shut down the labs, and like we have the samples, but we don't have DNA extract or anything yet because it was just, we couldn't do it yet. But one other thing too that just tied up with what Vinicius said, like whenever you're doing microbiome, it's important to remember that we are sequencing the DNA that's present in the sample. It doesn't mean that the bacteria were active at the time. It just tells us the DNA was there. So that's another thing, that's another nuance that we need to think about it. And there are different ways that we're using now to determine if they were active or not. So I don't want to like add too much noise and like those things like into this presentation, but those are other things that we're looking at right now to understand even better.
Okay, we have a couple of questions coming in to ask about whether any kind of testing is available that a farmer might be able to send in to learn about what kind of microbiome is on their farm, or whether farmers will be able to sample microbiomes on their own farms and then send them to labs for analysis and tracking.
Does anyone else want to take this, or?
I'm not aware of anyone.
Yeah, so right now I would say that microbiome is definitely in the research phase. In humans, for example, there are companies that say, you know, send us a fecal sample and we'll tell you whether or not you have a healthy gut microbiome. One of those companies is actually being investigated right now because they're kind of making some clinical claims that can't be made yet. So I would say right now, it's hard for us to even say, what does a healthy dairy farm microbiome look like? If it's anything like the human microbiome, there may be many different types of healthy microbiomes, right? So that's one really big challenge, is to determine what does a healthy microbiome look like and what does an unhealthy microbiome look like. It's somewhat easy to do with specific diseases like mastitis, but if you want to sort of say, do I have a healthy farm microbiome, that's very challenging. I do think in the future there will be clinical applications for this, where farmers and others can send in specific samples and actually get meaningful results from them. But I think that we're not there yet. So Luciano said, you know, that he's putting together protocols for how to collect milk in a way that's compatible with microbiome research. And there are definitely some additional considerations that have to be taken into account when you're collecting these samples. But we're not really there yet to say, to give sort of definitive answers on these types of things. It would be more of a sort of, oh, that's interesting, this is what the microbiome of my farm looks like.
Okay, yeah, so that you might want to be cautious of a claim of a company that would offer that service or something at this point. Yeah, okay.
Yep. Yeah, definitely.
Yeah. I think that kind of might help answer the next question that we got, which was, can we increase the good microbiome on a farm? I think it may still have to be determined what that actually means based on what you just said.
Yeah, I think definitely be on the lookout for our webinar too, because we're going to be talking a little bit more detail about things like pre and probiotics. And that's definitely microbiome science, and trying to understand the science and the evidence base behind those types of things. So I think that'll be covered in webinar 2 in a little bit more detail. Certainly people are trying to do their best to determine what are good microbes and how do they help. And so that's a big area of research right now.
Okay, here's another question. This is maybe too much of a random question, but do you all know of any research related to apple cider vinegar use in livestock water and how that affects dairy cow microbiomes?
I'm not aware, but I can speculate that it will definitely change it, because you're going to change the pH. So you change the population of bacteria that will be there. Which one, like naming bacteria that going to go up and down, that I can't.
And it would probably, you know, whether that it probably would change, you know, one of those microbiome compartments that we talked about, but maybe not the entire gastrointestinal tract or other things like that. So.
Okay, this question says, there's a lot of issues related to methane production, and there's the idea that with organic, less milk is produced per animal, meaning that methane production per pound of milk is potentially more. Is there any evidence that the rumen microbiome of organic cows is producing different amounts of methane?
So there is a paper. I'll go first because others on the call might have more information. There have been a couple papers looking at the rumen microbiome of high and low methane emitting cows. And there were some differences in the rumen composition of high versus low emitting cows. And I think these were cows that were being fed the same diet. So people are definitely looking into that and how diet, rumen microbiome composition impacts methane production. And there seems to be some role of the rumen microbiome in that relationship. Anyone else on the call have anything to add to that?
Okay. Also, another question, there's a really interesting figure that you put up from the study that looking at calf microbiomes over the first few weeks and then 1 and 2 years old. Is there any work that you're aware of that compares the microbiome of calves fed colostrum versus colostrum replacer?
I'm not aware of any off the top of my head. Anyone else on the call?
I was thinking about that question before, but I really couldn't come up with anything.
And they do have some some papers out, but that's more like on milk versus milk replacer. I don't remember specific on colostrum and colostrum replacer. But there are a couple groups that have been working with it.
Okay.
Yeah, so I guess on that, we're not aware of any specific papers for colostrum.
Okay. Are the healthy biotics in the udder the same as those found in calves' stomachs, or are they totally different bacteria that communicate with each other well?
Can you read that one more time, Alice?
Yeah. Okay. Are the healthy biotics in the udder the same as those found in calves' stomachs, or are they totally different bacteria that communicate with each other well?
Oh, that's a great question. I would say probably a little bit of both, but I haven't seen the only thing. So if I can, I'm just going to quickly go back to one of these plots. This one. So if you'll see, there's a lot going on here, but they did take on the lower left-hand part where it says maternal microbiota down in the legend, they did take colostrum samples in this. And then those are the little like dark asterisks in the figure. And then they took calf samples from the different parts of the calf microbiome. And so the closest to the mouth, assuming that these calves were actually nursing on the mother, which I'd have to look at the paper, but the pharynx is in the diamond like magenta color. And so you can see that the diamond magenta colors are up on the top part of the graph and the colostrum sort of on the left-hand middle side. So they sort of cluster separately from each other, indicating that there may be some bacteria in common between the colostrum and the pharynx microbiomes, but not a lot. And then you can see as you go down like the reticulum in the red triangles there, potentially a little bit closer to the colostrum. So again, this is a way to compare those microbiomes. I think probably there are big differences between what you would find in the calf stomach and what you would find in the milk or the colostrum. And there probably is also crosstalk between both the microbes and also the immune cells that get passed back and forth too. So it's very complicated, I guess would be my large takeaway answer, but no one specifically looked at that, I don't think. Anyone else know about that?
Okay, let's see. We have a listener from Uruguay who thanks you for the information. So thank you for joining us. It's exciting. Let's see. Going back to make sure I didn't skip some questions here. Okay, so so is the unique microbiome fingerprint similar to a DNA fingerprint?
So that's, no one can say for sure, but definitely there's a lot of DNA within all of these microbes and a lot of different genes. And if you were to be able to assemble them into some sort of data fingerprint, potentially they have uniquely identifying properties. And we don't know the level to which that happens. Actually, we have a separate grant from the USDA to look at microbiome DNA as a potential fingerprinting method. But there's a lot of computational work that has to be done to understand how uniquely discriminatory those fingerprints may be, and how we would assemble them sort of from a computational standpoint. So I think I hypothesize that it is like a DNA, like a person's DNA or a person's fingerprint, but it remains to be seen how powerful that will be.
Okay, let's see if anyone else has any more questions. We have just a couple more minutes left here. Let's see, going through here. Oh yeah, we had a question a little further back here about, can you talk more about organic opportunities? I'm not sure exactly what's meant by that question, but if that brings anything to mind about anything about how this specifically relates to organic agriculture or something like that.
I'm not sure if Brad is on the call, but he may be able to speak a little bit to just activity he's seen within the organic community in terms of microbiome. I don't know.
Sure. I think that some of this stuff may be very applicable to organics, especially when we talk about grazing and herds with different feeding opportunities, you know, going all grass-fed or not. One thing I think we also forget about is genetics. Breeds of cows can play a role in the microbiome. So I think those are two things that people are starting to explore from a microbiome standpoint in organics.
Okay, that's great. So I think that's it for the questions, and we're almost out of time anyway. So I'd really like to thank everyone for joining us today for this presentation, and to thank you all for your continued work in these very strange and difficult circumstances. Thanks to everyone. Thank you for the team of researchers, and thanks to all of the great questions, and to everyone for joining us, and I hope you all stay healthy.
Thanks very much.